Thank you so much for that extremely kind introduction, and thank you for having me here to share some thoughts with you. You can find all of the peer-reviewed stuff, the papers, the data sets, everything is at this address, and then here is a personal blog of what I think some of these things mean. What I'm going to talk to you about today is bioelectricity, but specifically the use of bioelectricity as an interface to the plasticity of the agential material that makes up your body, and I think this has massive implications for biomedicine going forward. I like to think of the end game of our field as something called the anatomical compiler. Just think someday you should be able to sit in front of a computer and draw the anatomy of the animal, plant, organ, biobot, whatever it is. You should be able to draw it. And this system will then compile that specification into a set of stimuli that would have to be given to cells to get them to build exactly what you want. Now, so in this case, I'm showing you this three-headed flatworm. But if we had something like this, then birth defects, traumatic injury, cancer, aging, degenerative disease, all of these things would go away. If you could convince cells to build whatever you wanted them to build, all of these things would go away. Now, why don't we have something like this? We're actually very far away from it. Where we stand today with molecular medicine is that we're very good at this kind of information, which cells and what the cells are doing, what individual proteins are binding to what other proteins, which genes turn on and off other genes. The molecular information is very strong, but we're quite a distance away from being able to restore limbs and repair birth defects and things like this. And so why is that? I'm going to argue that this is because where medicine is today is where computer science was in the 40s and 50s. In those days, in order to reprogram a computer to get it to do something different, you would have to physically interact with the hardware. Here she is. She's rewiring the machine to get it to do something different. And so, what I think we've just now started scratching the surface of is to understand actually the plasticity and more specifically the intelligence of the living material, meaning that it is reprogrammable and for the same reason that you would laugh if I told you today that on your laptop to switch from Microsoft Word to PowerPoint, you'd have to get out your soldering iron and start rewiring the thing, right? That's we don't do that anymore. Why? Because we understand that the material that we're dealing with is reprogrammable, and living material is that and more. So this is what we are made of. We're made of individual cells. So here's this is a free living organism called the lacrimaria, but it gives you an idea of what individual cells can do. This thing is incredibly competent in its own little sphere of influence here. There's no brain, there's no nervous system. It handles all of its physiological, metabolic, and so on, and needs all in one cell. And in fact, all of us were a single cell once. And these cells did this amazing process of embryonic development where they became one of these complex organisms, or even perhaps a human. And in fact, even below the single cell level, the molecular pathways within single cells already have learning capacity, right? The different signaling pathways and gene regulatory network components come together into a collective intelligence that is able to form six different kinds of memories, including Pavlovian conditioning. You don't need a brain, you don't need neurons, you don't even need the cell. Just the molecular pathway alone has this capacity, and we are now creating devices that we're using to train these pathways, and then there's many applications like drug conditioning and so on. So the idea here is that bodies are made of multiple levels, not just of different scales of organization, not just the nested dolls structurally, but actually every level has its own capability of solving problems in different spaces. So we're familiar with animals solving problems in the behavioral space, but your molecular networks, your cells, your tissues, all of them are constantly navigating these other problems. And we are made of this amazing multi-scale competency architecture. I'm going to give you just one example because time is short. I'm going to give you just one example of the kind of problem solving I mean. So this is a tadpole of the frog Xenopus laevis. You've got the brain here, here's some nostrils, here's the mouth, these are the eyes, and this is the gut. And so these tadpoles have to become frogs. In order for a tadpole to become a frog, it has to rearrange its face. So the eyes have to move, the mouth has to move, the nostrils, everything has to change. And you might think that this is a hardwired process, that somehow the genetics specifies that every organ moves in the correct distance, the correct direction, and then you get your frog. Well, we decided to test this because anytime you're going to make a claim about the level of intelligence of something, you have to do experiments. You cannot just assume. So we decided to test it. We made so-called Picasso tadpoles, where everything is scrambled. So the eyes on top of the head, the mouth is off to the side here. Everything is mixed up, like a kind of like a Mr. Potato Head doll. Everything is mixed up. And then what we found is something amazing, that these animals give rise to pretty normal frogs, and that's because these organs don't just move the right distance in the right direction, because then they would be wrong. They actually move in whatever way is needed through novel paths to get to where they're going, and they make a correct frog face. So this ability to reach your goal from different starting positions, right? Get your goal satisfied despite novelty, things that you did not know were going to happen, is a key aspect of intelligence, of intelligent behavior. And so now you have to ask a simple question: how do these cells and tissues know what a correct frog face looks like? Okay, how do they store the memory of the endpoint? And we know they can remember the endpoint because they stop when they get there. Okay, when they achieve the normal frog face, then they stop. So how do we actually, how can we think about a collection of tissues storing these kinds of memories? Well, we took our inspiration from what happens in the brain, which is a sort of familiar system where groups of cells store memories and guide behavior, in this case in three-dimensional space. And the way it works in the brain is this: you have a network of cells. So here's a neuron, it's touching this other neuron down here. It has these little proteins called ion channels, which let charged molecules in and out. And as a result, it acquires a voltage, and that voltage may or may not be propagated to its neighbors, and the flow of electrical signaling through this network is what underlies all cognitive activity. And so here is a video that this group took of a zebrafish, a living zebrafish brain. And you can see this amazing electrophysiology going on here. And it's the commitment of neuroscience that if we could decode this, so this is the project of neural decoding, if we could decode this, then we would be able to read out the memories, the goals, the preferences of this animal. And so that is what we would like to do, but outside the brain. We want to do it for the rest of the body. And the reason that's possible is because actually this amazing system of using bioelectricity to integrate information, store memories, and guide intelligent behavior is way older than brains. It, in fact, evolved around the time of bacterial biofilms. It's extremely ancient. And in fact, every cell in your body has these ion channels. Most cells have these electrical synapses to their neighbors. And your tissues are running these electrophysiological networks. And so you might ask the question: what do they think about? We know what our brain thinks about. What do the body tissues think about? And so I'm going to tell you that one thing they think about is shape. They think about arranging the body in the correct shape and then maintaining that against aging, against injury, and against cancer. And we can start so here, much like this video of this brain, we can start taking a look at the bioelectrical signals. This is a frog embryo. What you're seeing is a time lapse of a frog embryo, and you can see all the conversations, the electrical conversations that these cells are having with each other. Could we decode this? And here's what some of these patterns look like. First of all, this is something we call the electric face. So again, here's a time lapse of a frog embryo putting its face together. And there's a lot going on here, but if you look at one frame of that video, you can see that prior to the appearance of the craniofacial organs, this is the map that it's going to build. Here's where the animal's right eye is going to go. Here's where the mouth is. Here are the placodes. So this we are literally, what you are seeing here is literally reading out the electrical memories that tell the cells what a correct face is supposed to look like, and this is what guides normal development. So we can now begin to read out what it is that these cells remember as the correct thing to build. And not only does this bioelectricity serve as a kind of cognitive glue that binds individual cells into a global vision of what the whole large scale thing is supposed to look like, it actually does this for multiple embryos. Here, you can see if we poke this embryo, all of these guys find out about it. Okay, see this? So this injury wave. This bioelectrical communication tends to merge subunits into a coherent whole. And you can see here, these are individual cells. By the way that we're monitoring all of this is with voltage sensitive fluorescent dyes. That's a technology that we developed to use these dyes to help understand what the cellular collectives are thinking. Now, watching these patterns is all well and good, and in fact, you can use them to diagnose birth defects and so on. But of course, the more important thing is to start to rewrite these pattern memories. So if I'm telling you that these are memories, on the one hand, you want to be able to read and interpret them, and on the other hand, you want to be able to reset them for therapeutics. And in order to reset them, we don't use electrodes or fields or magnets. There are no waves, no frequencies, nothing like that. What we're doing is manipulating the natural interface that these cells are using to control each other. So on their surface, they have these ion channels which set the voltage, and then here they communicate that voltage to each other. So just like neuroscientists do, we take all of those tools and we can use pharmacology to turn the channels on and off. We can use optogenetics and so on. So we can control the voltage and the communication between cells. So I'm going to show you very quickly three stories that illustrate why this is powerful and why this is important. And they work not because we're so smart. They work because the system actually is using this electrical communication as a cognitive medium, as a decision making substrate for determining growth and form of the organism. So the first thing I'm going to show you is a quick story about cancer. So if we take nasty human oncogenes like KRAS and P53 mutations and so on, and we inject them into tadpoles, they will eventually make a tumor. But before the tumor becomes apparent, you can already see using this voltage imaging, you can already see that these cells have an aberrant voltage from their neighbors. And what happens is that voltage causes them to disconnect from the network. Once you've disconnected from the network, you can no longer remember this grandiose goal that you had before, where the collective was working on building and maintaining organs. As far as you're concerned, you're an amoeba. The rest of the body is external environment, and that border between self and world has shrunk. The cognitive light cone of that cell has drastically shrunk. It is not more selfish than other cells, it just has a smaller self. So, cancer is in large part a dissociative identity disorder of the cellular collective intelligence, and it happens by breaking these electrical connections. Now that weird way of thinking about it has implications. It means that instead of trying to kill these cells, you can actually try to force them to reconnect with a pattern with the other cells that are holding the memory pattern. And so we've done that here by co-injecting. So here you inject the oncogene, but you also inject an ion channel that's going to force the cell to be in the correct electrical state. And so these are the same animal, and so you can see the oncoprotein is blazingly strongly expressed. It's all over the place, but there's no tumor. Okay, and there's no tumor because it's not the genetics that drives, it's not the mutation that determines the outcome. It's the physiology and the cellular decision making. And these cells are remaining connected and they are working on the same goal as they were before, making nice skin, nice muscle, whatever. Okay, so that's a story at the level of single cells. Now I'm going to show you what this means for birth defects. So here's a brain of a tadpole, and you can see here forebrain, midbrain, and hindbrain. And what we can do is we can introduce a mutant to a gene called Notch, and that's a very important neurogenesis gene. And so these animals have a very abnormal brain. The forebrain is missing. The midbrain and hindbrain are a big bubble. You can see the difference. These animals are profoundly affected. They have no behavior to speak of. What we were able to do is to build a computational model of the electrical pattern that normally tells the brain what size and shape it's supposed to be. And we simply asked the question: given this kind of disorder, what channels would you have to open and close to get back to the correct shape? And the computational model gave us an answer. And we found a couple of drugs already human approved. So this is already already in use in patients for other reasons. But when you use them, here's what happens. Now you got a normal brain. They have actually learning rates indistinguishable from controls, and yet they still have that mutation. Okay, so this is an example, and I'm not saying this will always be possible, but this is an example of fixing what is fundamentally a hardware error, meaning the mutation. You fix it in software. You fix it by a computer designed brief biochemical intervention that resets the electrical patterns so that the tissues know what to do, and then they make a correct brain. And the final story that I'm going to tell you is about regeneration, limb regeneration. So adult frogs do not regenerate their legs, unlike salamanders. So if they happen to lose a leg, then 45 days there's basically nothing. So we asked the question: could we communicate with these cells through the bioelectrical interface and guide them towards the leg growing path in anatomical space instead of the scarring path? And so we designed a cocktail. Basically, a 24-hour application of that cocktail ends up with driving about a year and a half of leg growth. So at that point, immediately within the first 24 hours, you get the pro-regenerative genes coming on here. This is MSX1. You then, by 45 days, you've already got some toes, you've got a toenail, eventually a pretty respectable leg, and it's touch sensitive and it's motile. Okay, so again, keep in mind that in this case, we did not have to manipulate that process during that whole time. We didn't have to talk to the stem cells. There are no scaffolds here. We didn't have to micromanage it at all. We provided a very early signal that said, go down the leg building path. That's it. And so at this point, I have to do a disclosure because David Kaplan and I have a company called Morphoceuticals where we are trying to push this forward to biomedical use, is such that eventually, so now we're trying this in mammals, and so eventually you will have these wearable bioreactors, not just for the limbs, but potentially for all organs that would provide the correct bioelectrical payload in terms of ion channel drugs. So that would then trigger the growth. Not trying to control every aspect of it because you have no idea how to do that, but to provide a trigger stimulus. And the final thing that I want to show you is the ability to induce novel organs, just to kind of nail the idea that what we're doing here is reprogramming the pattern memories of these cells. So I showed you that little eye spot in the electric face, and we wondered what would happen if we reproduced that same pattern somewhere else. So what you can do is inject into this early embryo RNA encoding a particular ion channel that produces a little pattern of a particular voltage, and sure enough, those cells get the message and they build an eye. Okay, on in this case on the gut. These eyes have all the same layers, retina, lens, optic nerve, all the same stuff. And just notice what this means. First of all, it means that bioelectrics, of course, as I've shown you in these other examples, is instructive. It's instructive at the organ level. We did not have to say which genes to turn on. We didn't have to tell the stem cells what to do. We found a high level subroutine call that says build an eye here. Okay, and the cells are very competent at doing that. The material, as I was pointing out, is not only competent to receive low information content stimuli and then have a very complex downstream response, but also, it does this cool thing that other collective intelligences do. For example, ants. When ants come along a piece of food that's too big for them to move, what do? They recruit other members of the community. Well, these cells do the same thing. If we inject a few cells, they can tell there's not enough of them to build an eye. So what do? They recruit a bunch of cells from the environment that we did not inject at all. So there's the ability for the material to scale itself to the available to the message that they receive. And just to point out, if anybody's interested in plants, this is another great example of the plasticity of life. You might think that the oak genome encodes for this shape, because this is what you see most of the time. These acorns give rise to exactly this shape, this flat green thing. But along comes this non-human bioengineer, this wasp, which is a parasite, which puts down some signals for the plant cells, and it hacks them. It hacks the morphogenesis exactly the way that I've been showing you that we can hack the morphogenetic outcomes in animals, and it causes these plant cells to build this incredible spiky round thing, or even this stuff. So these are galls, and we would have had no idea what these cells are capable of if we didn't see this amazing example of a bioprompting or reprogrammability. And in fact, it's kind of interesting that the sophistication of what is built is roughly parallels to the sophistication of the hacker. So bacteria and fungi make these kind of boring, lumpy things. Nematodes and mites do a little better, but by the time you get to insects, you get this beautiful kind of construct. So this is what we're trying to do. Is it took millions of years for the wasp to be able to do this. We want to accelerate this process, and so the last example I want to show you is a new kind of technology, which are basically synthetic biopods. So when you look at this, you might think that this is something we got from the bottom of a pond somewhere, but actually, I could tell you that if you were to sequence it, it's 100% Homo sapiens. These are human adult tracheal epithelial cells. We have a process that allows them to reboot their multicellularity into this little creature. It's self-motile. Now, why is this interesting for biomedicine? What does this do? Well, and by the way, you would never guess that the human genome would make something like this. This is nothing like any stage of human development. Well, one thing they have the capability of doing is healing wounds. So if you make a bunch of human neurons in a culture like this, and you put a big scratch through the middle, right? So kind of a wound assay, and then you put these bots into their environment, they form this thing called a super bot cluster. And here you can see what they're starting to do. You lift them up 4 days later, you see that what they've done is knit the two sides of the wound together. So who would have thought that your tracheal epithelial cells that sit there quietly for a long time, just kind of getting rid of the mucus and so on, are capable of having a completely different life in a different form factor with the ability to heal some of your wounds. And this is just the beginning. This is just the first thing we discovered. They probably do a lot more. And so we're envisioning these anthrobots as personalized autonomous therapeutics. In other words, they're made of your own cells. If we inject them into your body, you don't need immune suppressing drugs. You're not going to reject them. They biodegrade within a few weeks. And in the meantime, there might be many applications in which these things could be cleaning out joints, looking for cancer cells, dropping off pro regenerative molecules, fixing up neural connections, and so on. And so the bottom line is this. Much of biomedicine today is focused around these bottom up technologies, so at the level of the hardware. But there's a whole a frontier of medicine that's opening up, which are these top down interventions that take advantage of the intelligence of the living material to not to micromanage molecular states, but to convince the cells to and reset set points. We can train them. Um, here are the electroceuticals that I've been showing you, right, which are basically just signals to get the bioelectric pattern memories shifted in the right way. And of course, AI is going to be very important in enabling us to communicate to all the different layers of the body. So my claim here is that future medicine is going to look a lot more like a kind of somatic psychiatry and not like chemistry, because the name of the game is going to be to really take advantage of the intelligence of these other layers of the body. And the final thing I will just say is that because of the plasticity of life and because of the innate problem solving capacities of tissues, pretty much any combination of evolved material, engineered material, and software is some kind of possible embodied mind. So, in other words, cyborgs and hybrids and augmented humans and weird, kinds of creatures that you could only begin to imagine. Some of these already exist. Many are coming. They will be here with us in the future, and that means that we need to let go of old categories around living things versus machines and all that, because the entire variety of life, what Darwin called endless forms most beautiful, are a tiny corner in the space of possible beings. And I think we need to take very seriously the idea that it's this technology is not just about fixing all of the medical situations that plague us today, but also to release a kind of freedom of embodiment where people really can reimagine their life in a very different way than the body they happen to have been given as an accident of the trial and error process of evolution. And we need to really work on a new way of synthbiosis of living up in a mutually beneficial way with beings that are going to be different from us. So I'll stop here. I'm just going to thank the postdocs and the students who have done the work that I showed you today. We have lots of amazing collaborators. I thank our funders who have supported this work, and the disclosures of these are the companies that have supported us over the years. So thank you very much, and I will stop there.